What we did in our study was to target both lentiviral and adeno-associated vectors to mouse CD8 to enable syngeneic mouse models, not only for in vivo CAR therapy, but also for other fields of gene therapy. And we selected a DARPin by ribosome display, and then inserted that DARPin into the entry machineries of both lentiviral and adeno-associated vectors and tested that directly on primary murine splenocytes. So this is all in vitro so far. And we found that both of those vector types, when targeted by the DARPin, are not only highly selective for mouse CD8, we're talking about selectivities upward of 95%, but that we also have excellent transduction efficiencies. The lentiviral vectors perform as well as human-targeted vectors would, and the adeno-associated vectors actually outperform untargeted AAV2 vectors by more than a factor of 20, which was very surprising because there's always the concern that these vector platforms, being based on human pathogenic viruses, don't really work with the mouse system. And we found that that's not really true. And by targeting these vectors to murine receptors, you can actually use them productively in the mouse system. As I said before, this is all in vitro for now. We just had a nice heading on a slide in the talk sessions that said in vivo veritas, and I think that's very true, and this is the next step for us, to evaluate these vectors in vivo. And since we're using syngeneic models which have a complete immune system, one of the major challenges will be to see how the host immune system interacts with our vectors and how it affects the effective on-particle dose.